What if a messy lab bench saved millions of lives? In 1928, a single, forgotten Petri dish would do just that, changing the course of medicine forever. A simple mistake became a miracle. Where microbes should have thrived, a strange halo formed, a zone of death where life was stopping life. One word, born from this accident, would soon enter every hospital on Earth: penicillin. This is the story of how an accident became a cure, and how that cure launched the antibiotic revolution. Our story begins in London, 1928, at St Mary's Hospital, Paddington. In a notoriously untidy lab, bacteriologist Alexander Fleming was consumed by a single problem. Having served as a captain in the Army Medical Corps during World War I, he had seen firsthand how simple wounds could lead to fatal infections. He was studying the enemies of soldiers and civilians alike, Staphylococcus bacteria. Day after day, he cultured these dangerous microbes, searching for a substance that could kill them without harming human cells. A magic bullet that existing antiseptics failed to be. In August, Fleming took a brief holiday, leaving a stack of uncleaned Petri dishes on his bench. It was a characteristically messy move that would have profound consequences. When he returned, he began sorting through the chaos. He noticed one plate had been contaminated. A spore of penicillium mold, likely having drifted in through an open window from a lab downstairs, had landed on the agar. But this wasn't just contamination. Around the mold, there was a clear, sterile halo. The Staphylococcus colonies, which should have covered the plate, refused to cross that invisible boundary. It was a distinct inhibition zone. The mold was secreting something lethal to the bacteria. Many researchers would have simply discarded the spoiled plate. But Fleming, with his prepared mind, looked closer. He recognized the significance of what he was seeing. He cultivated the mold, at first calling its bacteria-killing secretion mold juice. He soon gave it a more scientific name, derived from its source, penicillin. So how does it work? Penicillin belongs to the beta-lactam family of antibiotics, molecules built around tiny chemical rings with enormous power. To survive, bacteria build rigid cell walls using special enzymes called penicillin-binding proteins, or PBPs. These enzymes act like construction workers, linking materials together to create a strong outer shell. Penicillin's beta-lactam ring is the perfect saboteur. It binds irreversibly to these PBPs, effectively jamming their machinery. Wall assembly grinds to a halt, leaving the cell dangerously exposed. Without a complete wall, the bacterium can't regulate its internal pressure. Fluid rushes in, pressure rises, and the cell bursts like an overfilled balloon. The infection begins to fade. Fleming filtered the mold broth to capture this active substance. On lab plates it worked beautifully, wiping out a range of gram-positive microbes. But turning this mold juice into a medicine was another matter. The penicillin molecule was incredibly fragile and unstable. His extracts were impure, and he lacked the chemistry skills to isolate, stabilize, and concentrate it in sufficient quantities for human trials. In 1929, he published his findings. The paper landed with a thud. The world, not yet understanding its potential, barely noticed. For a decade, the greatest medical discovery of the century sat on a shelf, a fascinating but seemingly impractical observation. Then, as another world war loomed, a team of scientists at Oxford University revisited Fleming's forgotten clue. Pathologist Howard Florey led the team. Biochemist Ernst Chain, a refugee from Nazi Germany, brought the chemical expertise. And biologist Norman Heatley provided the brilliant practical ingenuity. Resources were scarce due to the war. They improvised, turning hospital bedpans, food tins and bathtubs into makeshift fermenters to grow the mold. Their process was a relentless cycle: grow the mold, extract the compound, purify and concentrate it, then test its potency, repeat and refine. In 1940, they achieved a breakthrough. Mice deliberately infected with lethal doses of Streptococcus bacteria were given penicillin. they survived. The untreated control group all died. This was the proof the world needed. Now, for the first human trial. In 1941, a 43-year-old policeman named Albert Alexander, dying from severe sepsis after a scratch from a rose thorn, received the precious drug. He rallied dramatically. His fever dropped, his infection began to clear. But their supply was tiny. After 5 days, it ran out. The team even tried to recover penicillin from his urine to re-administer it, but it wasn't enough. The infection returned, and he tragically died. The case proved two things. Penicillin worked, and shortages were deadly. Production had to be scaled, and fast. With Britain under siege, Florey and Heatley traveled to the United States. There, government labs and pharmaceutical companies like Pfizer and Merck joined the effort. Engineers cracked the problem of deep tank fermentation, using huge, oxygenated vats to grow the mold at an industrial scale. Output surged. By D-Day in 1944, enough penicillin was being produced to treat every Allied soldier in need. Supplies reached field hospitals, and mortality from infected wounds plummeted. After the war, it became available to the public. Infections that once killed, pneumonia, scarlet fever, syphilis, became treatable. In 1945, Alexander Fleming, Howard Florey, and Ernst Chain rightfully shared the Nobel Prize in Medicine for their collective achievement. From a chance observation to a global medical revolution, penicillin ushered in the age of antibiotics. Today, these medicines remain precious, but their power is fragile. Resistance grows with every misuse, a stark reminder that this miracle of medicine is a resource we must protect.